Okay, welcome to NSTA Engage Spring 21. My name is Heather Ryan and I'm the session's moderator. Chris Herman and Rachel Mosey are supporting us behind the scenes today, and Karen Avery and Dan Williams are our presenters for Modeling How Cells Talk, Making Sense of Synapses and Signals. Please use the chat to communicate during the session. And then this chat will become part of the recording that will be posted on the Spring Engage site tomorrow. During the session, Rachel will post links to resources, registration for your free student modeling pack, and the session evaluation. I will also monitor chat for your questions, and we will let you know if we have time when you can unmute and interact. It is now my privilege to introduce Dan Williams and Karen Avery. Excuse me. Dan teaches AP biology, anatomy, physiology, and marine science, and runs the science research program at Shelter Island High School in New York. Dan earned his undergraduate degree in biology from St. Joseph's College and master's from SUNY Stony Brook. Dan is a past teacher fellow at Brookhaven National Laboratory's Office of Educational Programs and the Cold Spring Harbor Laboratory's DNA Learning Center. Karen Avery has been a biology and microbiology instructor at the Pennsylvania College of Technology for the past 5 years. Prior to that, she spent 15 years in the high school classroom teaching biology, honors, AP, environmental, and applied biology. Karen earned her undergraduate degree in biology and secondary education from Lycoming College and her master's in biology from Bloomsburg University. And she is currently a doctoral candidate in STEM education at the University of Pittsburgh. Both Dan and Karen have been conducting workshops and trainings on our 3DMD materials for educators like you for many years. And they both served in leadership roles on CBM grants and summer courses. And they have an outstanding presentation for you today. And now let's begin with the program. Welcome, Karen and Dan.
Hi, everyone. We're going to get started with a little interview format. So I'd like to ask Dan a few questions. Dan, what makes a good model?
First, I'd like to say hi to everybody too, but
oh, sorry.
That's okay. Good models. Well, that's a really good question, and it means different things to different people. You want a model as something that you can touch, you can see, you can manipulate. Often we want our models to be colorful so they can be eye-catching. We want our students to get interested in them. So we want a model that students are going to be fascinated about. We want our models to be able to somewhat show what reality is. Kind of like a Matchbox car is like a car. I could run it down a ramp and it does what car things do. So we could talk about the car without smashing through our classroom.
We're going to be focusing on the Synapse model today. What can we learn from the Synapse model, Dan?
That's a really long question. So what we hope to be able to show is how cells can communicate with each other. But more than just a lot of us, or at least myself, I would draw these things up on a board and it would just be meaningless pictures that the students won't understand. Where here we have these models that you see something leave one cell and attach to another cell and can actually cause shape changes and communications and shape equals function as opposed to meaningless two-dimensional pictures on a board.
So this is pretty complex. What do you find the most difficult when you are teaching these cell interactions?
And there's a little bit of music possibly in the background as we're changing classes, as we're all in school right now. And these interactions between cells, again, it's too much vocabulary, too many words that we have where the models the students can see, they can play with, and they can manipulate that we would find a lot more tangible for the students as opposed to just vocabulary and 2D diagrams.
So how do multidimensional models help with this?
Because we can play. And that's what we hope that you can see over the next series of slides is we get a chance to play with these models. More importantly, the students can play. That's where the curiosity and the learning continues.
And the great big question for today, how can you show cell communication, synapses, disruptions of communications between cells, and signal transduction using these models?
And that's what we hope to show. So how do cells talk? If we take a look at this image right here, there's lots of different ways that we can communicate. And I'd like to highlight the one on the left-hand side first, is we see that we're communicating language with shapes of our hands communicating different words. And so much of biology is shape equals function. And so these models are about shapes. Things will fit into each other. Things will change the shapes of others. It's also in some ways about touch. We could see how the different neurons are touching each other or very, very close to each other. So it's that close proximity that we see within the synapses. But are messages permanent or are they temporary? We have Post-it notes. We have Snapchat type things. We send out a message and it's used for a time and then it disappears. You know, how do cells communicate with each other? Well, they use all of these types of things with touch, with shape, temporary messages, more permanent messages that we could see throughout all of these models.
So if you have the kit that you got, if you pre-registered for this workshop, you might want to have that at the ready. We're going to take a second to walk through both the student modeling pack and the Synapse kit.
Hi, I'm Karen, and I'm going to take you through the two kits that we're going to be interacting with today in our presentation. Let's start off with the Membrane Student Modeling Pack, which you probably have in front of you right now. And you probably found it fairly easy to construct. I think your students will too. You can see that we've represented the phospholipids individually on this mat and also added in some gated channels, some leaky channels, and some our iconic sodium potassium pump. In addition, your students will have individual phosphates for modeling membranes. I've colored the phospholipid head on mine red. It seems to help with that transfer of knowledge. The water molecules, I color mine also red in the oxygen region. Some people like to keep the foam that they came in so that they can keep track of all the different pieces. You also have some ions. Sodium ions, which are blue and round, and potassium ions, which are purple and square, nice for your colorblind students. I label mine as sodium potassium, but you don't have to. We've got some glucose molecules to cut out and the all-important ATP molecule with a removable phosphate group, because if you don't have that, your sodium potassium pump can't do anything, right? So now that you're all set up, your students will learn how to use diffusion, facilitated diffusion, active transport, and the coupling of those two, which I think is one of the difficult biological concepts to grasp. Once they have that, we can talk about how two cells talk to each other. And for that, Dan is going to talk about the Synapse construction kit. You can see here that we've got the phospholipid membranes all set up. We transferred from individual phospholipids to a model that I call the train track model of the phospholipids, because it takes a lot less time to put together than those individual phospholipids. And we can start to add in some of the gated channels that we need and the leak channels, in addition to our sodium potassium pump. So now we can add our ions and start modeling with just one cell what happens, but also we can show what happens between two cells. So here we have a presynaptic neuron and a postsynaptic neuron in the synaptic cleft here. This one is a model of a cholinergic synapse. You can see that we've got acetylcholine in a vesicle. We also have a channel that Dan's going to talk about and a little molecule called synaptogammon, some snap-in snares here that we'll talk about later. And I've added a receptor to the postsynaptic neuron. And so, and a little acetylcholinesterase, which is going to become important later. We can also build a dopaminergic synapse. And for that, we've replaced the acetylcholine receptor with a G-coupled protein receptor and added dopamine to our presynaptic vesicle. We can also build a GABAergic synapse. And so we've got a GABA receptor over here, and we've got GABA in the vesicle now. And these are all things that Dan can tell you a little bit more about later in the presentation. One of the nice things is that this kit also includes some ways of breaking this communication. So one of the best ways to learn about something is when it's broken. And also one way, a great way to figure out what your students know is to present them with a story or a problem that they have to use to work through a system. And so when we look here, you'll note that the kit also comes with sarin, so a little nerve gas, some nicotine, which the average teen is pretty familiar with. We also have propofol, which is a pretty quick acting anesthetic, some cocaine, and some toxins. So students can explore how these additional molecules can break or impact how these synapses work.
So if we start off with the basics like simple diffusion and osmosis and maybe even facilitated diffusion, those seem to be going pretty well in the classroom. But what happens when you get to the sodium potassium pump, or even the coupling between facilitated diffusion and active transport? So if you have your little kit with you, I'm going to talk over this video. One of the things that you can walk your students through is this sodium potassium pump. So you can see here that we're loading sodium into the sodium potassium pump and then phosphorylating it. Once this pump is phosphorylated, the phosphate is transferred, that changes the shape of the protein, which is going to eject those sodium ions into the extracellular fluid, the area, the interstitial fluid outside of the cell. So out they come. And it's nice to have your students set up a little chart of concentrations for the sodium and the potassium as they're going through this, so they can keep track of that building of the electrochemical gradient. Once the sodium is ejected, we have a place where the potassium is able to bind, and we only have 2 potassium for every 3 sodium. That's going to cause the kicking off of that phosphate group, which then allows the protein to go back to its prior shape, which is not, it does not have a high affinity for those potassium ions. So those are dumped into the cytosol. And you can go through that cycle a few different times and keep track of the electrochemical gradient as it begins to build. So as we move from that simple cell to the Synapse kit, we can start talking about how these parts all fit together. So in this video, which we can go ahead and start, you can see that we have a vesicle that is full of acetylcholine. And in order to make that vesicle begin to move towards the end of this neuron, we need a signal from calcium. That calcium is going to bind with a protein on the vesicle and stimulate that vesicle to start moving down some cytoskeletal fibers towards the synaptic bulb, where this connection, exocytosis, is facilitated by the connection between these snap and snare molecules. Once those snap and snare molecules kind of bind together, that is going to allow for that vesicle to fuse with the membrane on the synaptic bulb. And that's going to dump all our acetylcholine into that synaptic cleft. So right here, we're seeing some things that are usually left in the background, but my students are always asking me, like, how does the vesicle know which direction it's supposed to be going? How does it know it's supposed to be going towards the end and not back towards the soma of the neuron, and so on? So at this point, I've laid the foundation for you. Dan's going to tell you how he uses this kit to make the magic happen in his classroom.
So before I go into the voltage gated and ligand gated, we started out discussing what makes a good model. And when Karen was showing that sodium potassium channel, One of the things that makes this, or sodium potassium pump, one of the things that makes this so fantastic is its simplicity. She was able to go through all the different things, and I have my students actually draw those as exam images. So it's not just something that, oh, we demoed it in class, or, oh, they had a chance to play with it. It becomes a way that they can now see and express what's going on to be able to answer questions on an exam. And a similar thing is going to happen here with the voltage-gated and ligand-gated channels. I used to present this all the time, and students would hear these words, but they never understood what truly was the difference between it. And I'll let the video speak for what we're going to see, because a lot of these are a little bit hard to manipulate in the air, and we didn't want to have accidents like music coming into play, so we recorded these ahead of time. So thank you for your patience on that.
What you see up on the screen right now is a sketch of what I would do when I'm doing the voltage-gated versus the ligand-gated channels. Up top, you can see the voltage-gated channel, just kind of two little parallel lines with something that's going to swing in and swing out, whether it's open or not open. And when it's open, you can see the little blue dots coming through, showing how the sodium is coming through the channel. And when it's closed, obviously the little blue dots can't get through. On the bottom, you can see a ligand-gated channel. First, it's kind of squeezed in a little bit, and so the sodium ions can't get through. And then, of course, some ligand bonds into it, and it's open, and the sodium can come waltzing through the channel. This doesn't give a lot of great information about what's going on biologically. And so what's one of the powers of the Synapse modeling kit is that we can show these different things, what's happening, and get into discussions of why things are happening. So here you can see up on the screen, I have my image, my old-fashioned image in the upper right-hand corner, and in the lower left, you can see the Synapse kit. And we have all these voltage-gated channels in orange and in green, whether they're the sodium channels or the potassium channels, and even a red one for the calcium channel. Coming in on the left-hand side, right-hand side, you're going to see that we have this pulse of ions that are coming through. These are the sodium ions from the action potential. And as the sodium ions come in, they're positively charged. Positive charge is going to change the chemistry. And as they come marching in, the voltage-gated channels are going to have to change shape. They change shape because the chemistry changes shape. The chemistry changes and they change shape. And so we see this now in the next picture. The sodium channels, and I have, because the action potential is marched through, the sodium channels and the potassium channels start popping open. Ions can start flowing through because the positive ions are causing a shape change, causing the gates to open. We can see that within, you could almost model it as these positive ions come here, pull it open within a sense. In the upper right-hand corner, I've replaced the image with showing the process of the voltage-gated sodium channels, where up top it's showing how they're closed, and at the bottom of the image you see how it's open. It's a shape change. It doesn't really look like our models, but you can try to relate how it's just things shifting based off of the chemistry. When they're based off of the chemistry, sometimes there's a hole, sometimes there ain't a hole, and that allows the ions to travel through. Ligand-gated was always also something that was difficult for me. So ligand-gated channels we now see at the bottom. This is in the synapse itself. So this is not in the axon. And the ligand-gated channel is the acetylcholine channel over on the far left. And so first what you see is the acetylcholine is coming out into the synapse because we've had an action potential. They're being released from the vesicles. And Karen had shown that earlier. As they're released from the vesicles, they travel into the synapse and they bind into the ligand. When something binds in, so if I have a receptor and something binds in, by definition, it's going to change shape. As these bind into the receptor, it causes the whole receptor to change shape, enabling a channel to open. The sodium can come rushing through. And so we've activated this. Well, it's different. First, you had the voltage gated because it was a change of ions, positive ions coming in, pulling the protein into a different shape. And you can model that with the little foam pieces. And here we have the ligand coming in. They have to bind in. That enables a shift change as well. And so, of course, we see here within the last picture, the sodium comes marching in. We have done it. We've activated the next nerve, and the students can physically see the difference. It's no longer that silly picture that I have up in the upper right, where I just kind of describe what's happening. The students can actually play with, touch, see the difference of what's going on.
So we talked about earlier that one of the best ways to learn about something is to break it. How do we break it? We're going to take a minute to watch that happen in this video.
Equally important to understanding how a synapse works correctly is understanding what happens or how a synapse can be broken or the signal from one cell can be disrupted when it's being transmitted to another cell. So we're going to start off with, as you can see here, a cholinergic synapse, and we're going to investigate what happens when the cholinergic synapse messaging system is broken. So when it's working correctly, acetylcholine will bind with its receptor, which will allow that receptor to open and allow for sodium to flood into the postsynaptic neuron. Thus, the message is conveyed from the presynaptic cell to the postsynaptic cell. Also important is the ability to shut that off. So we have an enzyme here called acetylcholinesterase, which functions to shut down the signal by splitting acetylcholine into two smaller subunits, which can then be taken up back into the presynaptic neuron and reused as a signal the next time it's necessary. So that message has effectively been shut off by acetylcholinesterase. Now, what could possibly interfere with that? Well, in floats a chemical called nicotine. What you're looking at right now is nicotine, and nicotine can break the communication between these two cells by competitively binding with the receptor for acetylcholine, forcing it open even, in the absence of acetylcholine. So what you see here is the sodium ions diffusing through that open channel, even though acetylcholine is still in the presynaptic vesicle and it hasn't been released. So that message shouldn't be out there. And finally, we have a third situation here, a second way to break this messaging signal. So acetylcholine binds with the receptor and sodium ions are diffusing into the cell, transmitting this message. What if we have so here's acetylcholinesterase doing its job and shutting down the signal. And now we have a nerve gas coming into play. And instead of competitively binding, that's sarin, instead of competitively binding with the receptor, it is going to competitively bind with the mechanism for shutting off the signal. So it binds with acetylcholinesterase, thus blocking competitively acetylcholinesterase. And when acetylcholine needs to be broken down, it cannot be broken down. Thus, the channel remains open and the message continues to be conveyed to that postsynaptic neuron, even though it shouldn't. So let's look at the dopaminergic synapse here. And in this situation, of course, we have dopamine as our message and a G protein coupled receptor as the recipient, or the way the postsynaptic cell is going to receive that message. So here's dopamine binding with a G coupled protein receptor and the subunits of that G protein separating and conveying the message in that receiving cell. And usually the dopamine will be then retaken up by the message sending cell, the presynaptic neuron in this case, which will allow that dopamine to be reloaded back into a vesicle in that presynaptic cell, or the cell sending the message. This allows for the G protein coupled receptor to be reset. Here is, cue the evil music, dun dun dun, that is cocaine. Now that we have cocaine in the synapse, cocaine is going to interfere with the message by blocking the reuptake channel in the presynaptic neuron. So now dopamine will bind with the G protein coupled receptor, but rather than being taken back into the presynaptic neuron so that the message can be shut down or turned off appropriately, dopamine continues to be present in the synapse because it can't get out of the synapse, because cocaine is blocking the reuptake of that dopamine back into the presynaptic neuron so that the message can stop being transmitted.
Again, going back to our introduction, we had talked about what makes a great model. And one of the things that makes a great model is all the different ways you could apply it to new and interesting things. So on the next video, we're going to talk about using these models to show signal transduction, and where I tried to play and show other applications. When you have these, if you think outside the box, there's a million different types of cell communication that we could start talking about and start modeling. That said, in here, I'm going to be using an MDMA receptor that was part of a pilot kit that we had tried a while back that I don't think that's necessarily available. I left it in there because you can make it out of anything. When we do these things in classroom, I've taken pipe cleaners and pulled them into the models because they could work and the students get inventive. More ways to play, more ways to see, more cool things that you can get your kids involved with. And it's that cross-cutting concepts. If I keep using the same models with new applications, they start seeing cell membranes are used here, cell membranes are used there. Cell membranes are how we communicate between cells. So here you get to see my adaptations for signal transduction and some other areas.
So this next topic is one of the more difficult topics, at least for me, to teach within an AP biology class. I wouldn't do this in necessarily any of my intro bio classes, and that's signal transduction. Signal transduction is kind of one of these, there's a couple of boxes up on the screen, magic happens and stuff. So first, what you see up here is my standard synapse that I would draw up on the blackboard in class. You see on the left-hand side, the presynaptic nerve, vesicles dump in all of the neurotransmitter, they're picked up by the postsynaptic nerve. The second thing that you see down here is kind of a standard signal transduction type problem. The ligands now, that's going towards the postsynaptic nerve, is going to bind to some ligand receptor. And that ligand receptor is going to activate a box, activated protein, which is, by the way, going to now activate some other boxes. And these boxes are going to have some sort of amplified response, pedal to the metal type thing. What the heck's going on with any of this? It's not real. It's not magic. So to use our models, a little bit of review. First, we have the stimulus coming in on the presynaptic nerve, and we see that the calcium channels will open, snap-snare proteins are going to join up, and dopamine is going to be dumped into the synapse. And so kind of just breezed right through that, but don't want to do this as magic of, where is all this coming from? As the dopamine goes into the synapse, it's going to bind to the dopamine receptor. And the dopamine receptor is going to change shape. That's been a theme that we've been watching for. So it's going to change shape in that the G protein part of it is going to separate. We're going to have an alpha particle and a gamma and beta particle that are going to come off, or subunit, I should say. We're not in physics. And so here we can see the alpha subunit and the beta and GABA subunit are going to separate out. That's a very easy level. Now we're going to go into more of the signal transduction level, getting into more proteins are going to be involved, those activated proteins. So this next slide, what you'll see here is, again, my signal transduction on the upper right-hand side with boxes, and the models on the left-hand side where we can actually see what's being activated. The alpha subunit is going to go up and bind with adenylyl cyclase, which is up on the cell membrane. Adenylyl cyclase will now be activated and start taking ATP and cleaving it into pyrophosphate and cyclic AMP. It's going to do this 4 times. So we can see over the next couple of slides where we get 1, 2, and the cyclic AMP is going to start binding to the activated protein. Well, what is that activated protein? Protein kinase A. And a kinase is something that's going to phosphorylate things. And so we'll see that first cyclic AMP will bind, and then others start ganging up on it. And we wind up with 4 cyclic AMP are going to bind in. And you could see that in some of the diagrams. You could see 4 little phosphates come on. But here we're actually seeing how they bind in. Causes it to change shape. It bursts, it explodes open, at least within this model. And as it bursts open, it's activated now, activated protein, box little thing that we have on there, and it's going to do other things. We can see that it's traveling over to the NMDA receptor, which is a transmembrane protein that's going to allow sodium to come in. And here's where it's going to get a little bit dicey. And you have to kind of decide what you want to show. So in this picture, you can see how the phosphate is going to be binding into the NMDA, and it could cause a shape change. And you can kind of cheat a little bit and say, and now sodium comes rushing in, we've activated the next nerve. But if you want to go into that detail at least once, here's how you can do it. So we're zooming in, we have adenylyl cyclase. The big, huge honking thing in the middle is NMDA, and we have AMPA, and you could look up all these things for the time of this video. I'm just trying to do alphabet soup, but you have the AMPA receptor on the right. And all of these are going to be involved in putting the pedal to the metal. This is where that response, you see the 3 words up there, it's not just one response, it's a mega response of what's happening. So first you have the phosphorylation of the NMDA. Okay? Second thing you're going to have is amino acids are going to bind to the AMPA receptor. And the AMPA is now going to change shape, and it's going to allow a depolarization. We can see some of the sodium start coming into the cell. As the sodium comes into the cell, it changes the shape of the NMDA. Following all this, lots of little steps. NMDA is going to release the magnesium. You see it pop out into the extracellular fluid. And now it's also going to bind with a series of amino acids. Basically what we're doing is we're popping it open, slamming it open with the amino acids. And that's happening to both channels now. Why is this happening? In some sense, because the protein kinase A has phosphorylated. So one thing leads to another, signal transduction, and we now have both channels are open and everything starts coming in. Tons of sodium start coming in, pedal to the metal. And that's what you'd want to see. Do I really need the students to be able to identify every little step and know all the alphabet soup? No, but I do want them to see that it's not just magic boxes up here. We have a real thing that is pushing this down, which is going to make memories, which is going to make intense feelings. That's the kind of thing that's going on in your brain. That's why we have the long-term effects from this type of thing. That's what signal transduction does. For some fun, I went and decided that, signal transduction happens a lot, happens in the immune system. So you could see up here on the screen using the same stuff, I'm modeling what's happening between a T cell and a B cell and COVID virus. So COVID is hanging out between the 2 cells. Here we have the 2 immune cells are coming together, and the vesicle is opening up, and it's going to release the cytokines. Cytokines are going to come down in and bind to a G-coupled protein receptor. Same type of idea. Different receptor, same idea. And because the G-coupled protein receptor will change, the alpha subunit is now going to come over to, lo and behold, adenylyl cyclase. And adenylyl cyclase will start cleaving ATP into pyrophosphatase and cyclic AMP. And cyclic AMP is going to go to protein kinase A. Protein kinase A is going to get phosphorylated 4 times. If you look up in that diagram in the upper right-hand corner, you see a little teeny tiny protein kinase A with 4 P's on it. Now it's not magic anymore. It's a model that I could relate between different units. And the protein kinase A is going to burst open. It's going to become activated. As it bursts open, this time it's not going to phosphorylate different membrane channels. It's going to phosphorylate DNA. And as it phosphorylates DNA, the B cell is going to start cloning itself. And so you have the effector cells that are going on. Lots of things are going to start happening. But you can see how tag, you're it, the DNA is on. Cross-cutting concepts, not just this model goes into one thing. We can make relationships between different units to get students to see that this is how biology activates things, talks to things, all through one pretty cool model.
And within this model, I like to leave the models out long term. It's not something, oh, got to get this lesson done and get to the next model. And I'll leave these out on tables because we can play with them and come back. And when a student asks a question, it's let's see how we can do this within the model. And what I'm going to show you on this next video is questions that came from my students. These are not my questions, they're theirs. I do want to throw out that while I go to conferences like this, I often think, oh, I can just show that up on a screen. I could just display some sort of animation or whatever, and the students will get it. And I've done that before. But what I've noticed this year of COVID is we can't play with the models as much because of social distancing. So I've had to resort a lot more to videos and to demonstrating things. And I've watched my questions plummet. I've watched where it used to be a lot more interactive with the students, but nowadays they're kind of just sitting back and waiting for things to happen instead of playing. You need the hands-on. It generates a lot more questions. And as I said, these are from my students.
As we mentioned in the beginning, the quality of any good model is the question that it generates. It's not so much that the model looks nice, that's important. But if you put a model out and the student's like, yeah, whatever, then that's not the best model. But if the model goes out and the student's like, oh, wait, what about this and what about that? Then you start seeing that this model is something that you want to invest in and you want to use. And the Synapse modeling kit is one of those things that generates really good questions. So what we see on the screen here is the actual Synapse area with the ligand-gated channel on the left-hand side. And what you see up in the right-hand corner is some images from David Goodsell, who does cellular landscapes that are out there. So what's happened is the synapses the acetylcholine has gone through, opened up the ligand-gated channel, and the students would start saying, well, is that the only ligand-gated channel that's going to be there? Is it 1? Is it 2? Are there 5 ligand-gated channels there? How many are there? Is there a variety of ligand-gated channels? So for instance, this one's binding acetylcholine. Do we find others that are binding, say, GABA on the same exact area? What's going on with all this? The students want to know, is the model much more complicated or much simpler than what's there? So looking at the Goodsell images, we can see that it's really complicated. On the right hand, the blue side is the GABA synapse, and on the left-hand side is the acetylcholine synapse. And we can see similar structures in both, but it implies that there's different synapses, different nerves have different type of receptors. So we can model that, where we have one nerve with one type of receptor, one nerve with another. Then the students want to know, how do we reset? How do we clean up? In our modeling image, we have those little brown things which are showing the acetylcholine esterase. What this does is it breaks up the acetylcholine. Well, how the heck do we get them back up? Well, we have those yellow things there which are showing the reuptake channels, but is it an active transport? Is it a passive transport? Is it just for a matter of there's so much acetylcholine in the synapse that it diffuses back in? How does it get into the right vesicle? If there's going to be multiple neurotransmitters out there or multiple different chemicals that could be high in the synapse, how do we make sure it's going into the right place? The synapse snares have joined in to force the vesicle to actually dump out the acetylcholine. My students want to know, well, how does it go backwards? What's the reset button? And if your students are asking these questions, you might not have all the answers for it. That's okay, because this is showing your students are engaged. They're thinking, they're questioning. It's the wonder. We want to teach wonder in science. We want to teach questioning in science. We don't want memorization of facts. And so looking at this area, all the different ways that the students will question will show what kind of a great model it is. The second image that we have down here is a little bit further up the axon. And so I show a different model that 3DMD has, which is the actual nerve. And the students want to know, well, how do the vesicles get all the way to the terminal branches? The cell is huge. It's like 3 feet long. Why do we wind up with the vesicles down at the end of the terminal branches, but they're not all along the axon? What got them there? Students want to know, how do we make sure that there's transmission going from the nucleus 3 feet away all the way into the synaptic knob? How are we making sure that everything is coordinated, everything is in the right spot? By just laying out all these models, the students start asking these questions. They're curious, they're engaged, they're trying to see what happens. And then finally, the last image that I have here is in the axon itself. And the students wanted to know, is this why we have myelin sheath? To make sure that the sodium ions are kind of concentrated around the sodium channels and the potassium ions are around the potassium channels? Are there little things like hold in all the ions? So is there like a vesicle that's outside that's a power pack to make sure that it goes in? Or is this something that they just happen to be out there, and when the channel opens up, we have enough diffusion going through? How is the nerve set? Do we have everything in and around the sodium potassium pump shown there in gray? How do the things from the sodium potassium pump get back over to where the ion channels are? And if they're asking these questions, they're paying attention, they're seeing what the science is showing. And of course, you could look up some of these answers. You might not know some of these answers. Science might not know these answers. Our job as teachers are to inspire, to get them curious, and these models will help to do that.
So Dan's pretty humble, but he does great things in his classroom, and he drives his students to, after finishing up with the questioning, to make claims. And so what you see here, or who you see here, is one of Dan's students making a claim about how dopamine and cocaine may interact with the postsynaptic neuron or the presynaptic neuron. He's using some 3D printed models. You can find files for these various places on the internet and print them, but you don't need to have those. If you need any type of inspiration for a story that could go with this kit, I'm sure several are popping up in your mind. But on my next slide, I have a couple of different stories that you can use. So you can talk about how caffeine keeps us awake. There's a wonderful little TED Ed video about that, which my students just love. So caffeine is a great story to use. Another one is, of course, opioid addiction. And for that, I also used a TED lesson. Each of these are less than 5 minutes, but so rich with information to kind of get that storyline started. And a third is the story of Aricept, a treatment for some types of dementia. It interacts with acetylcholinesterase in the synapse. And so I have a Wikipedia link that you can use. The other name for this is donazepam. So, oops, sorry, donepezil. I just get those last two syllabuses or syllables. Goodness, you can tell it's the end of the semester. Confused. So at any rate, what we'd like to do now is see if you have any questions for us about these kits and how we use them in our classrooms.
Thank you, Dan and Karen, for a great session. And please go ahead and post questions in chat, or you can go ahead and turn on your mic and ask us and ask Dan and Karen directly. And while we are waiting for that, I will just go ahead and share some of my closing information, and we can actually stay on for a few minutes after the session to answer any questions that come up. So once again, thank you for attending. And I just wanted to go over a couple of things that Dan and Karen used a Synapse construction kit today. And some of you had the membrane student modeling pack, and that's what we will ship to all the participants. So make sure you click on the link to get your free student modeling pack. And you can use the code 3DMD Engage 21 to get 20% off most of our products. And please click on the link that Rachel's posting for the session evaluation. And I hope that you can join us for our next session. And that is going to be, we're going back to Chris Chow, and she is going to be presenting making connections between DNA and chromosomes. And then after that, we have Karen and Dan back again, who are going to be focusing on our dynamic DNA kits. And that's, your title, Inspiring Student Questions with a Twist with DNA Modeling, if I'm remembering correctly. It's on a screen on the other side of the room for me right now. So thank you, everyone.